So what we're going to be going over today, and we won't finish it today, and we'll finish it up on Monday, are essentially the parts and functions of all the organelles within the cell. All the stuff we labeled yesterday, we're going to talk about what each of those does, okay, for the cell. That's going to be important because obviously your quiz on Tuesday will be a labeling quiz, and you won't just be asked for the name of the structure, but you're also going to be asked for the function of the structure. So you're going to need to know both, and that's what we're going to be going over today. Okay. First off, know the significance of compartmentalization. That's going to be an important term. Okay. Compartmentalization is essentially this. There are many compartments within the cell. Each organelle is its own little room. Okay? Each organelle has its own membrane that separates it from the rest of the cell. Right? That means it can have whatever conditions are best for whatever process it carries out. Right? Obviously, for the lysosome, okay, there's digestive enzymes in there that are breaking down food. And what is your food made out of? What do we eat? Okay, if you don't know what you eat, I don't know how you're sitting here. What do you eat? Okay, what's food? What's food? Food comes from animals and plants, okay? It's made of cells. Do you want the stuff in here that's capable of breaking down cells to be everywhere? No, because it'll do exactly what it's supposed to do, break down your cell. Okay, we don't want that. We want a separate compartment for that. Okay, a special room where that only occurs. Okay, same for the Golgi apparatus. We don't want all the waste products of the cell going everywhere. That's why we have a garbage man to take care of it. Okay, same with the mitochondria. Okay, it has a double membrane. In fact, it has to insulate itself against the rest of the cell because breaking down glucose to release energy releases a lot of heat. Okay? And it needs to insulate the rest of the cell against that heat, otherwise the cell will get too hot and other processes won't work properly. Okay? So each organelle has its own compartment. That's what compartmentalization means. Okay? Know the role of each organelle and understand that cells are a complex system of interdependent processes. Okay? Don't confuse that with independent. Independent means they just do their own thing and don't depend on anyone else. Interdependent means they are crucially dependent on each other. Right? Every process within your cell is dependent on another process within your cell. Right? That's what interdependent means. Okay, and understand how the cell carries out its functions. So we'll talk a little bit about transport, okay, as one of the functions, but obviously that's going to be point number 5. Okay. All right, so what does compartmentalization mean? Okay, it means that cells are divided into compartments by membranes. Okay, the mitochondria is a separate compartment within the cell. The nucleus is a separate compartment within the cell. Okay, it's obviously very important for the nucleus to be its own compartment. What's in there? The DNA. If the DNA gets damaged or broken or in some way affected, that could affect how the plant or animal is able to produce things. Okay, so this compartmentalization is only present in eukaryotic cells. Okay, the picture there is a eukaryotic cell. Plants and animals are eukaryotes. The organisms you looked at under the microscope the other day, the amoeba, the paramecium, the euglena, those are all eukaryotes because their cells all have organelles. A bacteria is a prokaryote. It doesn't have a nucleus. It doesn't have organelles. All the processes just take place all in the same place. Okay? Is that going to be as efficient? What do you think? Having a whole bunch of processes take place in the same place that maybe are quite a bit different, is that going to be more efficient or less efficient than having their own places to do that? Way less efficient. Okay? The example here is like your home. Within your home, there are probably 3 separate temperature environments. The environment in which you operate and live. It's usually kept around 21 degrees Celsius. Your refrigerator, which is kept around 3 or 4 degrees Celsius. Why do we want our fridge to be cooler? It makes our food last longer. If we leave food out on the counter at 21 degrees, it will spoil a lot faster than it does at 3 or 4 degrees Celsius. The third temperature environment is your freezer. That keeps food for even longer, right? It's about minus 15 or 16. Okay? But obviously it's not 21 degrees. Okay? If I had to find a happy medium between those three processes, I couldn't have three separate environments, I would have to keep my house at about 8 degrees Celsius. So I'm going to be cold all the time. My food's going to spoil. And anything I had in the freezer is going to spoil way faster. So is it doing any of those jobs really well? No, finding a happy medium never pleases, okay, I shouldn't say that, never pleases anyone, but it never really pleases anyone, okay? Because everyone had to give something up. And so nothing is working at its peak efficiency, right? If we allow each thing to have its own little room, it can have the perfect conditions in there. All right, does that sort of make sense? Okay, so the importance of compartmentalization is that it provides different local environments that allow the cell to perform functions not compatible at the same time. Okay, this is why all, without exception, all multicellular organisms are eukaryotes. Okay, there are no multicellular prokaryotic organisms. Okay, prokaryotes without these organelles cannot become specialized. Okay? And specialization is the key to being a multicellular organism. Okay? So you'll never see any really big prokaryotes. In fact, prokaryotes are all basically bacteria. Okay? Now, do bacteria have some things that they can do that we can't? Okay? Well, there are actually some things that a prokaryote can do that we can't. For example, we have found prokaryotic organisms in places that we would have thought should be completely sterile. Okay. We have found bacteria growing inside of geysers in Yellowstone National Park, where the temperatures are 200 degrees. Okay. And yet these organisms survive. All right. No eukaryotic organism could possibly survive in an environment like that. Okay. We found them in places that should be too salty, okay, too alkaline, that's basic, okay, too acidic for a eukaryotic organism. But because they're so simple, okay, they can survive and tolerate some of these conditions that we simply couldn't. All right, so the compartmentalization of cells has allowed cells to become larger. Okay? Eukaryotic cells are almost always bigger, okay, and more specialized in their functioning. Okay, so compartmentalization allowed for specialization. Okay, if you look at this here, to give you a size comparison, this is an animal cell, and it doesn't even fit in the picture. Okay, and here's a bacteria. Okay, this bar here represents a half a micrometer. So you're looking at a bacterium who's maybe only a micrometer and a half in size, whereas the animal cell is probably 50 to 100 micrometers. Okay, much, much bigger. And then your virus is obviously way smaller than that. Okay, questions there on what compartmentalization is? Usually a multiple choice question on that on the test, so something to know. All right, DNA is used to make materials. We talked about that yesterday. DNA is crucially important. Okay, your genetic code, okay, the order of your base pairs, okay, in DNA is what tells your body how to do things. Okay, in the same way that zeros and ones, okay, binary code tell a computer what to do, your DNA with A's and T's and C's and G's and their order of those tells your body what to do. Okay? It's that basic code that's essentially written in there. So in the environment of the nucleus, DNA in the form of chromatin, that means it's not in chromosome form, it's just spread out everywhere. Okay? Believe it or not, DNA has zero organization inside the nucleus. It would be like someone took a ball of string, completely unraveled it, pushed it into a pile and jammed it inside. Okay? That's what it looks like inside the nucleus. So remember how I said yesterday it was a library? Not like an organized one. Like somebody went in there and tipped over every single bookshelf and all the books are on the floor and then a tornado went through. Okay? So pages of books are everywhere. Okay? That's more what it looks like, unfortunately. The nucleolus is amazing. It can still find what it needs in that mess. It's like the hoarder who knows where everything is in its house. Okay? Well, actually, they don't. That's why they find like dead cats in the hoarders' houses and stuff. It's really gross. Okay, but yeah, it can find anything in there. Okay, no matter what, even though the DNA is kind of everywhere. All right. So what does the nucleus do? Well, the nucleus stores DNA. Okay. Inside the nucleus, protein synthesis is controlled, but it is controlled by the nucleolus, not by the nucleus itself. Right, so looking at this picture here, we've got kind of the artist's conception of what these look like on the left, and then these are electron microscope pictures of the nucleus on the right at various magnifications. So the nucleus, okay, looks like this, and we can see that, you know, it's fairly, it looks like it's got little bumps all over it, and it does. Those little bumps are the nuclear pores. Okay? They're basically little openings, just like the pores in your skin, that DNA can get in or out of the nucleus through. Nothing gets through the membrane of the nucleus. Okay? It only gets in or out of the nucleus if it goes through the nuclear pores. If you blow it up even more, you can actually see that the nuclear pores kind of look like a hole or an opening. Okay, that's what DNA and RNA can get in and out of. Right. The structure of the membrane itself, you can see this kind of crisscrossing effect. It almost looks like denim. Okay, and that's the actual interlocking of fat molecules, okay, to make up the surface of the nucleus. Okay. Questions on what the nucleus does? Okay, so the nucleus, if I wanted to give a two-word answer for what the nucleus does, it does, what does it do? Stores DNA, perfect. Okay, two-word answer, like if you were on a quiz and you had to label and give the function, nucleus, stores DNA, good enough. Okay. All right, how does this process work? DNA has information encoded in it. How does it get read? How does it get turned into materials? This is not a process I'm ever going to ask you to write out or describe to me in detail, but it's a process you need to at least have explained to you so you know what the importance of DNA is. Right? So inside of your nucleus is where your DNA is. That's the blue stuff here. Okay? DNA is a double helix. You guys heard that term before? Right? It's got two strands. Right? Because the strands are complementary, that means so if I have a strand of DNA, okay, A always goes with T. Okay, these are chemicals that are called base pairs. Okay, A always goes with T. C always goes with G. It's the order of A's and C's and G's and whatever that essentially codes for information. Since A always goes with T, I only actually need one side to know what the other side would be. So when your nucleus is copying DNA, it isn't copying both sides. It only copies one side because that's all that's actually necessary. Okay? You know the other side, if this was an A, has to be a T. Everyone follow me there? Okay, so what your nucleolus does is it finds the section of DNA and then it has it copied by a chemical called RNA polymerase, which means it makes RNA. Okay. Once that RNA is made, which is just a copy, single strand of DNA, it goes out through the nuclear pore, which we can see right here. Okay, so it goes out through the nuclear pore and it finds a ribosome, those tiny little things that are on the rough ER. The ribosome can read it. Okay, so the ribosome is like the foreman of a construction site. It has the blueprints, it knows how to read them, it knows, okay, who on the crew is capable of doing what job, and it makes sure everything gets done. Okay, so it reads the genetic code and it starts calling for the raw materials that are necessary. Okay, so those raw materials get brought in, okay, kind of like this. Okay, they get put into the ribosome and they get attached to whatever protein they're making. So this is a protein, this purple thing. Proteins are made out of amino acids. That's what these little individual purple parts are. There's a whole bunch of different amino acids. That's why they're different shapes. Okay? So all those amino acids come in. The ribosome puts them together in the right order according to what the DNA says. And then when it runs out of instructions, it's done. Okay? Now the protein is built. Right? So the steps are really DNA gets copied. Copy goes to the ribosome. Ribosome builds the protein. Okay, simplified greatly, it's like 3 steps. Okay, everybody follow there. All right, so the nucleolus produces RNA. Okay, that's its job. Okay, and in doing so, essentially controls protein synthesis. Okay, nucleolus also makes ribosomes. Okay, the ribosomes can actually get out through the nuclear pores as well. Okay, and so that's one of the things that the nucleus makes is these ribosomes. Okay. So if the nucleolus makes ribosomes, and all the information is in the nucleus, why don't we make the proteins in there? Wouldn't it make more sense to not send it outside and just do it all in there? Okay. The reason it gets built out here, two reasons. First off, there's more raw materials out here. Okay. It's hard to get stuff into the nucleus. We don't want to be bringing stuff in there in order to build something, because then we might bring in something bad. Okay. So this process always occurs out here to protect what's inside the nucleus. All right, so that heading, in case I ever ask you what DNA is used for, it's used to make materials. Okay, inside of a cell, there's obviously lots of cytoplasm, there's lots of little organelles and things like that, okay? But a lot of the internal volume of the cell is made up of smooth ER and rough ER, because it's an important way to transport things. It's like a highway, right? With lots of cargo moving along it. Okay, so it has to go kind of everywhere in the cell. So there is a lot of it. Okay, under an electron microscope, this is what the rough ER looks like. Okay, so here, like this here would be a piece of the rough ER, and every little dot is a ribosome. Okay, and we can actually see kind of over in this area here that there's some dense, kind of cloudy stuff in here. That would be proteins being built by the ribosomes. Okay. Over here is the smooth ER. So the rough ER looks fairly organized and linear, and the smooth ER is just kind of everywhere. Okay. And there's no ribosomes over there either. Okay. So that's kind of what it looks like. Okay, so the endoplasmic reticulum. In the smooth ER, there's several things that are getting done. Lipids are synthesized. What are lipids? Fats, yeah. Okay, fats are really, really important. Okay, now I say that, I don't mean go home and eat like a big jar of bacon fat. Okay, you shouldn't do that. Our diet as North Americans contains way more fat than we actually need. Right? We need fat. Make no mistake. If you're not eating fats, that's not good. You need to have fats in your diet, because that's what all your cell membranes are made out of. Okay, keeps your skin healthy, things like that. But you don't need to consume as much as obviously the average North American consumes. Okay, so the lipids are synthesized, and those are used for building or repairing membranes. Okay, complex carbohydrates are broken down. Okay, complex carbohydrates are things like starches. So if you had spaghetti last night, okay, you had a lot of starch, because pasta is mostly starch. If you had a whole bunch of rice last night, rice is mostly starch. Okay? What starch is a whole bunch of simple sugars chained together. So it would be like one starch might have anywhere between 5 and 20 glucose molecules in it. Okay? So does it contain a lot of energy? It certainly does. Okay? Those need to get broken down into shorter sugars so that your mitochondria can use them, or your lysosomes can break them down further. Right? So complex carbohydrates get broken down there. Okay? Complex carbohydrates are high energy. Fats are high energy as well. That's why French fries and potato chips are really calorie heavy, because they're starchy stuff boiled in fat. Okay? So that you're combining the two highest energy molecules together, okay, when you're making something like that. Okay, another thing that goes on in the smooth ER, poisons are detoxified. Okay, so any toxic chemicals that you may have ingested, okay, I mean in small amounts, obviously if you drank a big vial of poison, you're probably going to die, but okay, if you just had small amounts of toxins, and lots of things we eat all the time are toxic, your smooth ER is responsible for trying to break those things down. So in your body, which organ would have cells containing the most smooth ER? Your liver, yeah. Okay? Your liver is responsible for detoxifying your blood. So the liver cells are specialized to have more smooth ER than any other cell in your body. Okay? Because that's what your liver does. Breaks down all the nasty stuff that we consume. Okay? So that would include things like alcohol. Okay? That's a toxin. Okay? I mean, it's not outrightly poisonous, but in large amounts can be. Okay, it gets detoxified by the smoothie R. Aspartame, okay, would be another one. Okay, if you drink like diet sodas or diet drinks and things like that that contain aspartame, aspartame's kind of toxic, and it gets broken down there as well. Okay, so lots of things that we eat are really a little bit toxic and have to be gotten rid of. Okay, hormones. Okay, hormones can get produced there. Okay. So it essentially has like 5 jobs. How many would I expect you to list on a quiz? 2. Okay, if it's got lots of functions, give me 2 of them. Okay, that'll be good. Okay, the reason all this stuff takes place in the smoothie R, well, the lipids part anyway, the smoothie R goes everywhere in the cell, which means it's in contact with all organelles, and all organelles have a membrane that needs repairing. It's in contact with the cell membrane in lots of places, and that always needs repairing. Okay, as well as it isolates the toxins. If they're trapped in the smoothie R, they're not going out and about in the cell poisoning other organelles. So it kind of limits any damage to an organelle that can readily repair itself because its job is to repair membranes. And so it kind of makes sense to kind of keep it all in one spot. Okay, lipids, like we said, can be quickly moved to the needed sites. Poisons and drugs are detoxified quickly and removed through the cell membrane. Okay, travel within the ER limits contact with the rest of the cell. All right. Now, sorry, we never mentioned what the rough ER does here. So for rough ER, rough ER simply transports proteins. It's covered in ribosomes. The ribosomes are always making protein. The rough ER's job is just to move that stuff around. Okay, Golgi apparatus. We said yesterday that Golgi apparatus is like the garbage man of the cell. Its job is to package waste materials and send them to the membrane. Right? The way it does that is that waste materials come in on one side of the Golgi and they leave on the other. Okay, so it's got like a shipping side and a receiving side. So stuff comes in on one side, it goes out on the other. Right, so it comes in, and all the Golgi really does, it doesn't really do a lot of chemical alteration. That would have been done by the smoothie R already. All it does is package it in a vesicle, which is just a little membrane enclosed bubble. Okay. That membrane enclosed bubble is obviously made of the same material as the cell membrane. So these little bubbles will just, wander off and eventually run into the cell membrane. And when they do that, they just merge. Okay. It becomes part of the cell membrane, and whatever was inside just pops out. Okay. Everybody with me there? Okay. It would be like if you had, it's a little bit gross, but if you've ever walked your dog and you have to clean up after your dog, right? You put the little glove thing on, okay? You pick up the poop and then you turn it inside out, the poop's inside, okay? Well, if it's closed up, that's like a transport vesicle. And then instead of just throwing the whole bag away, which is what you should do, okay? It would be like if you turned it back inside out and pushed the poop out, okay? Then that would be more like what a transport vesicle and the Golgi apparatus does. I can never think of a better analogy than that. I'm sorry, it's kind of gross, but it wasn't as bad as the athlete's foot thing yesterday, right? Okay. All right, so that's what the Golgi apparatus does. Under an electron microscope, this is what the Golgi looks like, okay? This over here is the receiving side. So you can see all the stuff coming in is small, okay? Very, very small, because it hasn't really been packaged in any way. It's coming from the smoothie R and other places in the cell. What the Golgi does is it makes those vesicles. Like here's one right here that's just being formed. And here's a whole bunch of them on their way out to the cell membrane. Okay? So you always on one side of the Golgi have big bubbles, and on the other side you just got small dots coming in. Okay? Those small dots get packaged, each in their own vesicle, and those go out to the cell membrane, and they're gotten rid of. All right, so that's how the Golgi works. Okay, so Yeah, so you can just say wastes, okay? Contents of a vesicle are modified for transport out of the cell. We could say gets rid of waste. Okay, the lysosomes. Now, lysosomes are only present in what kind of cells? Animal cells or the cells of chemoheterotrophs, because they would also be present in a fungus. Okay, so we have food coming in. Okay, food comes in through the membrane through a process called phagocytosis, which we'll talk about later. It's basically just the enveloping of food. Okay, gets put into its own little vacuole or vesicle, and that vesicle will meet up with, this is actually labeled incorrectly, okay, with a peroxisome. Okay, so the peroxisome has got the digestive enzymes in it. When it merges with the food vacuole, then it becomes a lysosome, and digestion occurs. Okay, the chemical breakdown of whatever was in there. This can also be done with cellular structures that have ceased to work or have become damaged. All right? When that happens, it's called autophagy. Okay? It means eating yourself. Okay? If an organelle stops functioning properly, the cell is the ultimate recycler. It'll just go, well, you know what, it would be like, well, that shed fell down. But you know what, some of the wood in it is still good, so we'll take it apart and salvage what we can. Okay? That's what your cell is going to do. It's going to go, well, this mitochondria stopped working, but we'll take it apart, still got lots of good fats in it and things like that. We can reuse that. Right? So it digests it, essentially uses it as food, and some of those raw materials might be used to build other things. Okay? Everybody with me there? Okay, so that's what the lysosome does. Basically just digests food, right? So if we wanted to say, like a short kind of description, it would be this: digests food. Okay, they do have a very strong membrane, obviously, because we don't want the stuff that's in there to get out. In fact, the lysosomes are a way for the cell to protect other cells within the organism. Okay? Your cells have a pretty intricate mechanism for detecting when a cell has gone bad. Okay? And that happens sometimes. A cell gets damaged and it starts behaving dangerously. Right? Cells can usually detect if that has happened to them, and if they detect that that's happened to them, they'll kill themselves. Okay? In order to save the rest of the organism, they will destroy themselves, and the way they do it is they rupture all their lysosomes. Okay? And so all the digestive enzymes get out into the cell, and they digest the cell, okay? And it kills itself. Right? So that mechanism works most of the time. As you age, that mechanism breaks down, and it doesn't work as well. Okay? Which is why you are typically more likely to get diseases like cancer later in life. Okay? As that process breaks down, your body doesn't detect that it's not working properly. Okay? The most common thing to happen to a cell is its DNA gets damaged and it starts to divide out of control. Okay? That's what cancer is. It's your own cells dividing out of control. Okay? That's why usually you detect cancer as a lump. Okay? Wherever the cancer is located, you get a tumor. Okay? That tumor is your own cells, but they've just started dividing and dividing and dividing. Okay? And what happens is you get this huge mass of cells, and they're taking all the nutrients and energy from the surrounding cells, and that essentially stops the other cells or starves the other cells. So if that happens in your liver, you get a tumor in your liver, the parts of your liver that are working properly are starved of materials, and you get liver failure because of this tumor. Okay, does that kind of make sense? Right? So the reason your body doesn't do anything is because it doesn't see anything wrong. Those are your cells. Okay? And so it's, well, I'm not going to send the immune system after those cells. They belong to us. So it doesn't attack it. Right? And that's why most cancer treatments involve killing rapidly dividing cells. They'll help get rid of the tumor. So if you're taking chemotherapy, okay, if you've ever seen someone who's taking chemotherapy, they lose their hair, they lose a lot of weight typically. That's because the cells that rapidly divide in your body are the ones that produce hair, fingernails, and stomach lining. Okay? Those are all rapidly dividing cells, and when you're taking chemotherapy, all those cells essentially are killed. Okay? Your hair eventually grows back because you stop taking chemo. Right? Radiation, same thing. They focus a beam of radiation on the tumor and kill those cells by essentially frying them. Okay? So all those processes get rid of it. Okay, and that's of course if the mechanism that's supposed to detect it isn't working. Okay? Most of the time your body detects that there's a problem and those cells kill themselves. All right, the mitochondria. Okay, the mitochondria is where cellular respiration takes place. Okay, that reaction we showed you yesterday where glucose reacts with oxygen and produces carbon dioxide and water. Okay, that occurs within the mitochondria. It also releases a lot of heat, which is why the mitochondria has a double membrane. It acts as an insulator and protects the cell from the heat that's generated by that process. Okay? So we'll say, as kind of a quick summary of that, we would say burns sugar for energy. Okay? You could also say cellular respiration. That would also be a good answer. Okay, so under an electron microscope, this is what a mitochondria looks like. You can see inside there are all these folds, all these layers. Kind of the artist's conception is kind of showing those. Okay, the reason there's all those folds is it increases the surface area, which means there's more places for those reactions to take place, so they take place more intensely. Right? Or just more surface area usually means more of it happens. Okay? And the more of it happens, the more energy can be released. All right, questions on the mitochondria? Okay, chloroplasts, present only in plant cells. Okay, and if I was writing on a quiz what a chloroplast does, I would probably write 3 words. Carries out photosynthesis. Okay. Okay. I would definitely include photosynthesis in my description of what a chloroplast does. Okay. The reason that chloroplasts are there is it concentrates the chlorophyll, among other photosynthetic pigments, but okay, primarily chlorophyll, into small places as opposed to having it just floating everywhere in the cell. It's much more efficient to have it concentrated. Okay. It'll generate a lot more sugar that way. Okay. Another thing is it's got a double membrane, just like the mitochondria does, because anything that absorbs solar energy is going to get hot. Okay, I mean, imagine like a black car and a white car side by side on a sunny day. Which one's going to be hotter to the touch? Black, because it absorbs more solar energy than white. White reflects a lot, black absorbs a lot. Okay, any organelle or anything that absorbs a lot of solar energy is going to get hot, so there's an insulating double membrane there to protect the rest of the cell. Okay. Other organelles, cell membrane, okay? If you're going to write something for the function of the cell membrane, it should be what I just underlined there. Controls endocytosis and exocytosis, and use those 2 words, okay? I don't want to see controls what gets in and out. That's the scientific way of saying that. Endocytosis and exocytosis. Okay? Cytosis refers to movement in the cell. Okay? Endo is into, exo is out of. Okay. Now I told you the other day that the cell membrane is far more complex than most diagrams show it to be. Okay? This is actually what the cell membrane looks like. It's not like the sheet of cellophane like it looks on your diagram. Okay? It's a matrix of a whole bunch of things. Right? So primarily, the membrane is made of fats. Okay? That's what all these little things are here with the tails that go down. So it's actually a double layer of fats. Okay? And the fats point tail to tail in here. That's what essentially separates inside from outside. Okay? Because fats and water don't mix. So this essentially keeps fluids from the outside out and fluids inside in. Okay? There is some movement back and forth, but it's limited. Okay, other things that we find in there, okay, would be proteins like this one, this big coily thing here, okay, is a protein. There's glycoproteins, there's receptors, there's big globular proteins like this, okay, they're all lodged in that matrix of fats. Okay. What a lot of these do is they act as antennas for chemical signals your body can send to cells. Okay, so you would have several receptors on every cell membrane that would receive adrenaline. Okay, when adrenaline hits the membrane, it would trigger that antenna. That antenna would signal the cell, adrenaline is here, you know what to do. Okay, and when your adrenaline hits, your fight or flight response takes over. The metabolism of your cells radically changes. Okay, you're able to do things you normally wouldn't be able to do. And like there's all those stories, like the little old lady who lifts up a tractor off of her husband, things like that because your adrenaline is going. Okay, I mean it's not always that incredible, but everyone has similar reactions. Okay, when your adrenaline starts going, okay, cells in your body all react differently. Okay, one of the big things. Increased blood flow to muscles and brain, okay? Because that's part of your fight or flight response. You need more energy, more oxygen to the muscles that are going to get you out of that situation and to the thing that's going to think of a way to get you out of that situation. Blood flow is usually taken away from the digestive system, okay? Which is oftentimes why when your adrenaline is kind of starting to pump, you often will feel a little bit nauseous, okay? You kind of get that feeling in your stomach of, oh God, okay? Well, that's because blood is actually being drawn away from the digestive tract to feed your muscles, your heart, your lungs, and your brain. Okay? So those are all reactions that it would have, and each cell would have receptors for that. Okay? Same with, they would have receptors for insulin, which would tell them, oh, hey, insulin is here. That means there's too much sugar in the blood. Let's start absorbing some. Okay? And then your cells would absorb more sugar through their membrane. Okay? That sort of makes sense? Okay, so the membrane is far more complex than what we saw earlier here. Okay, cell wall, present only in what? In plants. Okay, and the cell wall is made of cellulose fibers, and it's the frame. It keeps the shape of the cell. Does it control what gets in and out? Well, here's a colored electron microscope picture of the cell wall. The purple stuff is the cell membrane. This is the cell wall. It's what? It's hollow. Okay? The cell wall is exactly what as described here. It is a frame. Okay? It would be like if you've ever seen a house being built. Okay? When they first put the walls up, there's only the studs. Right? You can walk through the walls. The walls are there, but they're not enclosed yet. Okay? That's what the cell wall is. It's just a frame. It keeps the shape of the cell, but it's full of holes. Okay? Anything can get through the cell wall. It serves no protective function whatsoever. It's just there to support the shape. Okay? And that's what we can see here. And it's made of cellulose, plant fiber, dietary fiber, stuff that keeps you regular that we can't digest. Okay? We as humans have no enzymes, no digestive chemicals that can break down cellulose. Okay? So if you eat celery, you're not getting anything out of it. Okay? You might get a few vitamins, but okay, you're not going to get in very much energy from celery. That's why it's one of the things that's always listed. If you're going on a diet, eat lots of celery. It fills you up, but it doesn't give you anything. Okay? You can't digest it, so it goes through you pretty much unchanged. Okay? Corn, same thing. Okay, goes through you pretty much unchanged because it's made of mostly cellulose. Right? Everyone follow on that? Okay, so we can't digest that stuff. If you're a herbivore, cow, deer, moose, whatever, you do have enzymes that are capable of breaking down cellulose, but their stomach is also a lot different than ours. Okay, all those herbivores have a 4-chambered stomach. Okay, there's different digestive processes that go on in each part, and then they usually puke up part of their food, chew it again, and swallow it. Yeah, it's pretty disgusting, actually. It's called. If you've ever seen a cow chewing their cud, that's what they're doing. Okay? They've barfed up a little bit of the grass they ate, and they chew it again, and they got that gross drool coming out. Okay? And they swallow it, and they digest it. Okay? Lots of herbivores do that in different ways. Rabbits eat their own poop, and then they digest it a second time because it doesn't get fully digested the first time. Okay? If you believe in reincarnation, don't come back as a rat. You have to eat your own poop. It's disgusting. All right, so cell wall, does that make sense? Okay. Okay, water vacuole. Right? The water vacuole uses what we call turgor pressure to support the cell. Okay? And turgor pressure is an important term. We haven't talked about it yet, but we will. Okay? It's basically just that water pressure from within. Right? So the water vacuole is like air inside a tire. Okay? If you don't have enough air inside the tire is flat and it doesn't support the weight of the bike or car or whatever it's on. Okay? When it is full, it exerts lots of pressure and it can hold lots of weight. So that's what the water vacuole does. When it's full of water, it pushes on the cell and makes it stiff. All right, and lastly, centrioles. Okay, so centrioles aid with cell division. Okay, that's their job. I drew a picture of it yesterday, but my artistic ability is not very good. Right, so blow this up a little bit so we can see it a bit better. Okay, so when a cell is getting ready to divide, okay, it takes the centrioles, and the centrioles actually also divide. They go to each end of the cell and they grab onto your DNA. That's what's going on in this picture here. They've got fibers that are grabbing onto each chromosome and pulling them apart and making sure they both end up in the right places so each cell gets the right amount of DNA. Okay? Those are only present in animal cells because only animal cells are going to have radical shape changes. Plant cells are not. Okay, and microfibers and microfilaments. We called that the cytoskeleton yesterday. Okay, they help the cell stay intact during changes in shape. Okay. Questions on any of those? Okay, it's a lot to review. Good strategies for this kind of stuff is to make up like flashcards, okay, or things like that, because it is a lot of memorization. So it's going to be a lot of, I got to read over my notes, I've got to review this stuff. Okay, making your own kind of jot notes, kind of like what I did here, and I said, this is kind of the important part. You might want to make little jot notes like that at home. Okay, that's another time you process that information, more likely to remember it that way. Okay, so little things like that can go a long way. Or just having a friend and quizzing each other back and forth with the cell diagram or whatever. Okay, so we'll start on transport. We're probably only going to get through diffusion here if we're lucky enough to get through that. Okay, cells need to transport a lot of material just within. I'm only talking about within the volume of the cell itself. Okay, the primary method by which that is done is diffusion, because diffusion is free. Okay? It is a passive process that occurs naturally. The cell doesn't have to do a thing to allow diffusion to transport materials. Okay? However, as you are probably aware, you typically get what you pay for. Okay? If you've got you go to the app store on your phone and there's like the free version of the app and then the paid version of the app. And the paid version of the app is almost always better, right? Because they just want to get you in with the free one. Okay? Well, with diffusion, you get something out of it. You get stuff transported from where there's lots of it to where there's less of it. And it's free, but there's problems. It's slow. Okay? It's like, getting something sent to you courier post, okay, or like with UPS, or getting it through the regular mail, right? One is really slow, but it'll get there. The other is a lot faster, okay, and gets there obviously more quickly. So diffusion works kind of like this. I always describe it like a fart, okay? If somebody farts, eventually everyone in the room is going to smell it. If you are close to the person who farted, the smell is more intense than if you are further away from the person who farted. Okay? Why is that? Because the further the fart molecules have to travel, the less concentrated they become. Okay? And they are traveling by diffusion. They are traveling from an area of high concentration to an area of low concentration. And after a few minutes, okay, they'll have diffused to the point where nobody can smell them anymore, okay, because their concentration will have gotten so low. Everybody with me there? Okay, that's how diffusion works. So you've got spots in your cell that are producing certain things. There are spots where they are needed, which means there's probably not very much there. So stuff will naturally move from high to low. But it might go other places as well. And the cell really has no control over that. That's another drawback to diffusion. It doesn't specifically go where it's supposed to. Okay? It'll probably end up there, but it's not always going to end up just there. Okay? So diffusion is slow, and it's not targeted. Those are two problems with it. But it's free, and it generally gets the job done. Okay? That is why it is the most commonly used form of cell transport. Okay, and that's what we see with that picture there. Ink is just being put into water, okay, and we see that it's eventually getting less and less concentrated and spreading out, okay, over the entire area. All right, so underline that. And this. Those are the two important things about diffusion.